Welcome to our first Evolution and Cancer chat. Today we'll be talking about Alex Cagan's work looking at mutation rate and lifespan. James, I find this work really fascinating because it suggests there might be an important link between somatic evolution, the mutations that can lead to cancer, and aging. And I'm really looking forward to chatting with Alex Kagan and our two discussants, Zach Compton and Don Lemaine, about this paper today.
Yeah, me too. It was my favorite paper of the year. I think it really adds some fundamental insights into several, really critical questions in the fields of both cancer and aging. For example, there's the well-known Peto's paradox, where different animals of hugely variable size, varying in size by millionfold, like from a mouse to a blue whale, and varying in lifespan by up to 100-fold. And yet basically all of these different animals seem to have essentially similar risk of getting cancer, which is relatively low during their natural lifespans.
Right. So the paradox is like, if every cell every year has the same likelihood of getting cancer, then why aren't large organisms getting way more cancer than smaller organisms, right?
Exactly. And it really speaks to another fundamental, which is sort of understanding life history theory. How is it that different animals, in response to different sort of environmental needs and threats, evolve different lifespans and different body sizes, right?
Yeah, and also different sort of strategies about how they develop and grow, and kind of like the risks that they take as an organism, both in terms of their behavior and in terms of, do they grow really fast because they need to be big in order to be successful, or do they sort of grow more slowly and conservatively? And there can be lots of trade-offs with that, right?
Exactly. No, the trade-offs. I mean, do you take the live fast, die young lifestyle like a field mouse, where your external environment is so hazardous that to invest in the long game would have been a wasted investment? Or are you big and strong like an elephant and you can take the long game? And it's not always being big. I mean, bats by flight have avoided predation, and naked mole rats by living underground have avoided predation. So big isn't the only strategy, but it is a common one for long life. And this paper really adds some fundamental insight into at least one of these mechanisms that helps these organisms probably avoid both cancer and aging during the period of their life when they're most likely to be reproductively successful.
So it really kind of ties together a lot of threads about human health in a framework that can help to unify some things, it sounds like.
Exactly. The fields of cancer and aging can no longer really be considered separate fields. They're really intertwined. They go hand in hand.
And this issue of mutations, right? Cell mutations that happen over the lifetime, that's one of the really key pieces that helps to connect everything.
Yes, no, absolutely. I mean, mutations are clearly one of the key factors. We're learning more about them in terms of both how they contribute to cancer, which I think has been long understood, but also learning more about how the DNA damage contributes to our aging. And it's not just by sort of disrupting genes. There seems to be effects on the epigenome and sort of on the structure of our genome in addition, that sort of just makes it not function quite as well as we get older.
Yeah, I mean, to me, the question of, how do mutations over the course of the lifespan affect all aspects of our health and well-being, not just cancer, that's been something that's been really interesting to me for a long time. So seeing Alex's work on this, it was really exciting to, see some mechanisms that can help to really sort of unify this approach to human health that thinks about, sort of what's going on by considering sort of the evolution of organisms as well, right? It's like, so it's to me, one of the things I love about the field of evolution and cancer is that, well, I love evolution, right? I mean, so do you. You've got, your Charles Darwin in the background there. You've got your, fish with legs, right? We love evolution. And with cancer biology, it's like double evolution, right? You've got the evolution going on inside the body, the somatic evolution, the cells evolving over the course of a lifespan, which can lead to cancer susceptibility. And then you've got the organismal evolution, right? The way that populations of organisms are evolving to suppress cancer. And what we see in terms of cancer susceptibility is a combination of what happens when you overlay both of those things.
Right. And understanding how different animals have evolved this ability to avoid cancer during their natural lifespans is, really going to provide fundamentals that should be informative to human health. And I think that's always something that I've thought is a cool thing about studying things from that evolutionary perspective, because you are truly studying the fundamentals. You're not studying something that's only relevant, to in one context. If it's relevant across all mammals, for example, it's probably super relevant to us.
Totally. Well, James, I know somebody else who is just as excited as you and I are about this paper. Zach Compton, should we bring him on the stream to chat with us?
Let's do it.
Hey, Zach, how's
Hey guys.
it going?
Good. How are you?
Good, Zach. Where are you? What are you up to right now?
Yeah, I just finished my PhD. I'm now a postdoc at the University of Arizona. And I've really been focused on comparative oncology throughout my PhD. I mean, now as a postdoc, I'm kind of focused on tumor evolution, but there's been so much exciting things going on in comparative oncology. I was like, let's reel back the postdoc a little bit and focus on comp onc a little bit because there's just so much going on here.
So, Zach, what did you find so exciting about this paper?
The first one fundamentally that was exciting to me is as you have your head down during your PhD, Alex's paper comes out and you have to take a step back and be like, wait, we didn't know this? Like this kind of fundamental truth that kind of Alex was able to elucidate about mutation rates. I was like, I'd assumed that this work has already been done, and it's such a nice narrative. I mean, just fits perfectly into evolutionary prediction, and to see that outlined and that no one had done this before, I mean, it's just incredible.
Yeah, just the sheer breadth of mammals that they looked at. I mean, and you always wonder, how did they get these, right?
Yeah. I mean, nothing will make you feel more hopeless as a scientist, especially as a young scientist, to look at the work that Alex and Indigo did and be like, how will I ever come up with that?
Yeah, no, it's a really beautiful study that I think in some ways it hadn't been done. It couldn't have been done 5 years ago.
Right.
The technology simply wasn't there for the sort of the sequencing requirements. But the beauty is just the breadth of it and the carefulness of it, and then just, of course, the elegance of the results.
Yeah, I mean, just 2 decades into kind of the genome era, and it's amazing the volume of sequencing that had to be done to achieve what Alex did. So it's just incredible.
Yeah. All right. Should we go ahead and bring on Alex? I think
let's chat with Alex.
Yeah, I know all of us. We can't wait to actually have Alex on here so he can tell us exactly what he found instead of us like going on and on, right? So
yeah, gushing.
Alex, Kagan, welcome.
Hi everyone. What a great introduction. It's great to be here and talk about this research. Yeah, it's been a fantastic journey.
Yeah. So how did you get to where you are now, Alex?
I guess I'll cut to the recent years. But so I come from a comparative genomics background. So looking at population genomics, looking at different traditionally like great ape species. I was very interested in what can we learn from evolution. But I always thought of it in terms of comparing individuals within populations. And it never crossed my mind, as I think it still doesn't cross a lot of geneticists' minds, that there's actually this whole evolutionary world going on within our own bodies. And I think a lot of that, as James mentioned, is because the technologies just weren't there. We didn't have ways to look at these mutations that aren't shared across all our cells. They're just single or a few cells that have these mutations. And I heard about the work that was starting to go on at the Sanger Institute as I was finishing up my PhD. And I thought, wow, this new whole landscape of unexplored evolution within the body is just starting to be explored. And we already have all this great theory about population genetics, evolutionary biology that's been developed over decades before we even had genomes. And it's just ready to be applied to learn about what's going on within the body. So I think it's such an exciting time where we have this great theory and now we have these new technologies, and what can we learn by combining them together?
Well, Alex, I just have to ask too, you're an amazing illustrator. You illustrated my book, which was phenomenal. And like it has visual thinking and like visual representation. Has that contributed to kind of the way that you look at this field of evolution in cancer?
Absolutely. I think so. Often I find genetics is quite challenging because it's an invisible world. We still can't even see mutations. We infer them from sequencing data. And especially with somatic evolution, you have these clonal landscapes, you have cells competing with each other. It's very spatial. I think you kind of have to think about it in 3 dimensions. So I think visual dimensions in illustration really does help to get your head around what is otherwise a very hard kind of field to imagine, because we can't see it, so we have to find ways to visualize it.
Speaking of which, should we show some of your figures and some of your illustrations? Are we ready to get into that?
Sure, let's do it.
Okay, awesome.
Yeah, so this is kind of the key kind of first figure of the paper. So we were very interested, just to go back a step, we'd looked at the Sanger at somatic mutations within humans, and we'd learned that we are accumulating these mutations as we age, but we had no idea outside of humans really what's going on. And so what we wanted to do was to take a range of mammalian species with different lifespans, different body sizes, and see, are they accumulating somatic mutations at the same rate as humans, at a faster rate, at a different rate? And are there any kind of life history variables such as lifespan or body size that explain differences that might exist in somatic mutation rate? And so what we did is we looked at 208 intestinal crypts. We chose the same cell type because we know from humans that mutation rates can vary from cell type to cell type, which itself was an amazing discovery. So we looked at intestinal crypts, worked with London Zoo and others to get 16 mammalian species. And to isolate these crypts, we did laser capture microdissection. So we made histological sections, put them under the microscope, and we isolated individual intestinal crypts from all of these species, and we sequenced them with a low input genome sequencing method that I mentioned earlier, kind of enabled us to start to look at somatic mutations. And no one had done this really in other species. We had no idea if it was going to work. And you can kind of see here at the top that when we look at the number of somatic mutations we're seeing, it varies a lot from individual to individual. So at first we're like, oh, is this just noise? Is this not working? But then you can see in the bottom panel that if you actually correct for the age of the animals, in all of these species, we're seeing this linear increase with somatic mutations with age, as we've seen in humans. So that really gave us confidence that actually this method is working, and we're able to see this accumulation of somatic mutations with age.
Alex, so then I have to jump in with a question. Yeah. You said nobody done this before, so you didn't know if it was actually going to work.
Yeah,
was that scary to be like, we're going to take on this huge project and we have no idea whether it's going to work?
It was scary. I guess I was kind of naively excited. I remember it was a very exciting day when we got the first data from a Siberian tiger that had died of old age at London Zoo. And we looked at the spectra, which we'll come onto in a moment, and they matched so beautifully what we see in humans. And you really couldn't explain that from an artifact. That was when we knew, okay, this is going to work, and then let's just get as many mammals as we can. So that was an exciting day.
Awesome.
Yeah, and then so if we look at the next slide, so something else that's really cool you can do with this data is you can do more than just look at the number of mutations, because we know that different kind of processes that cause these mutations leave distinct signatures in the DNA in terms of where they occur in the DNA. So for every mutation, you can look at the base before and after it, and that gives you this kind of trinucleotide context for every mutation. And that enables us to kind of do a form of genomic archaeology. So we can look at the mutations, see where they occur, and then from that infer what are the processes that cause those mutations. And so we did that for each, kind of pooling all of the mutations we had for each species. And when we do that, we see broadly, these are these kind of plots of looking at the types of mutations you see. Across all these species, you can see they're broadly similar. We see a lot of these C to T mutations, but there is some variation. I mean, here you can see this kind of in some species like mouse or ferret, you have more of these these C to A mutations. And so we were curious about these differences. And so on the next slide, we kind of explored that. And what we found was that they're all mammals. So perhaps unsurprisingly, it's the same 3 mutational processes that are active. And these are all endogenous cellular processes. So they all happen just naturally within the cells. It's not due to exposure to pollutants or other environmental factors. But kind of what we didn't expect and what was surprising is that although the same 3 processes are active, the relative amount at which these processes are active varies kind of consistently from species to species. So just one example is that in mouse and in ferrets, you can see this kind of higher proportion of purple on these plots. That means what we infer from this is that they have a lot more oxidative damage, so oxidative stress causing mutations in these species. And this is kind of one of those results in the paper that we see it. We have no idea why that is. So is there something special? Are laboratory mice more prone to this kind of stress? Do they have issues with how they repair it? But these are kind of raising these hypotheses for future studies. Like, why is this the variation in activity of different mutational processes? What are the consequences for cancer risk or aging? But before this, we had no data on this. So we're kind of just very keen to generate the data to describe what's actually going on within somatic cells within different species. And hopefully, as we have more species that maybe share certain life history parameters, we can start to understand what are the underlying causes for this variation. And then going on to the next slide, the kind of kind of the key take-home message of the whole paper that we were undescribably excited when we found this result, is that we wanted to see, are there any life history traits that explain the differences in somatic mutation rate that we see? And what we found, kind of strikingly, is that lifespan really explains somatic mutation rate across mammalian species. So 1 over lifespan explains over 82% of the variation that we're seeing across in mutation rate across these species. And kind of the simple way of the take-home message really is that despite having very different lifespans, all of these species are ending their lifespan with a similar number of somatic mutations. So a mouse that lives for only a few years, its mutation rate is just clicking at a much higher rate than a human. So that when a mouse reaches 3 or 4, it's got the same number of mutations you'd see in an intestinal crypt in a human that's 80, 85 years old. And strikingly, what we didn't see, which we were kind of expecting we might see, is that there's no impact of body size on this. And that's interesting because of this concept of Peto's paradox, that these large, long-lived species are able to live so long without cancer, and how can they do that? So we thought we might see an effective lifespan and body size. The longer you live and the more cells you have, evolution has to lower the mutation rate. But really all we're seeing is that it's lifespan that dictates that. And that suggests that somatic mutations are having some role in mutation rates, having some role in aging and also cancer resistance. But there must be some additional mechanisms for these larger bodied species that explains how they're able to resist cancer. So kind of a striking result, I think, but also raises all these interesting questions that we don't currently have answers for. And so, yeah, that's just kind of the take home from the paper.
That's really fascinating. We have a question from the audience here right now, or a comment really. I wonder if you multiply mutation rate versus the proportion that are from a particular mutational signature, you might get a stronger association with cancer. Did you look at anything along these lines, Alex?
Yeah, it's an interesting question. So yeah, I have to think about that a bit more, but we were very interested in because we have these, we can associate mutations to particular signatures. And so we could kind of say, okay, X percentage of mutations come from oxidative damage, X come from other processes. Do some of these correlate better with lifespan than others? And for that, we found no, it's actually the combined number of mutations that matters, suggesting that evolution doesn't really care where the mutations come from. It cares how many you have at the end of lifespan. That's what selection is kind of acting on. But we haven't looked in terms of actual cancer rates. So that would be something else. It'd be interesting to explore that.
That's fascinating. And I mean, it's just wild to think that across so many species, basically all organisms are dying with approximately the same rate of mutations in their cells. I mean, it's really mind-blowing, like that kind of like as a general thing.
Yeah, that's exactly what we think. It's crazy. We almost couldn't believe how strong the association was, but it raises so many questions. So this is just mammals. This is just one cell type. So if we look in other cell types, well, we see the same relationship. And also, does it hold in birds, in reptiles? We just don't have the data, but we know that these are the kind of questions that we want to answer.
Okay. Yeah. Carlo Maley again is saying that the ferrets have one of those signatures and have a very high cancer prevalence in general.
Yes, that's a good point. So I think absolutely kind of the direction of future travel is to look at these, this is just 16 kind of almost randomly picked species from what was available at zoos, but to really systematically look at species with low versus high cancer prevalence would really help us tease apart maybe some of these signatures like oxidative damage are more likely to be associated with cancers. I think that's a great idea.
Well, and I just want to invite everyone who's watching, please add your comments, add your questions as we go. And right now I'm going to bring Zach back in because I know that he has some really burning questions for you, Alex.
Thanks, Athena. So actually looking at your slides just today, a question that popped up that has nothing to do with cancer is, a central kind of question in evolutionary medicine broadly is what is the so-called natural lifespan of humans? And looking at the collection of species you analyzed and humans being the longest lived species among those species. And it looks like we have evidence that natural selection may have intervened to kind of constrain somatic mutations. What insight does that give you, if any, on what might be a more natural lifespan for humans? That it might be a little bit longer than some of the hypothesized ones in the literature.
Yeah, that's a great question. It's challenging with the data that we have because human was by far the longest lived mammal.
Right.
It kind of the slope kind of fits to what we have from human.
Yeah.
So definitely, I mean, I'm currently in the process of trying to get samples from really long-lived species, because what the data suggests, which is really exciting, is that species that have longer lifespans than us should theoretically have better mechanisms of DNA repair than we have, but we've never actually been able to measure somatic mutation rates in these longer lived species. And if we can do that, maybe we can start to learn, well, are there certain processes that they suppress? What are the mechanisms? And that's where I think there's a really exciting future potentially for increasing the human health span or reducing cancer risk, is kind of learning from the solutions that those longer lived species have already evolved. And it's kind of really frustrating that we didn't already have, like a sperm whale or a bowhead whale.
Right.
I'm just desperate to look at somatic mutation and verify the prediction that they will have a lower mutation rate.
Yeah, I mean, you bring up a good point that, like, in comparative oncology studies, and I mean comparative studies broadly, even with the incredible sequencing technology you have at your fingertips at the Sanger, one of the harder parts is just sample acquisition, is trying to track down and shipping these samples all over the world. So I mean,
exactly, and it's really, absolutely 100%, and it's really, we want to know the age of the animals at the moment for when they've died, so we can calculate that mutation rate. And when the whales wash up on the beach, they don't have a birth tag on them. So we're trying to look at these other. There are ways you can try and calculate the age of animals. So we're exploring all these avenues to try and get that data.
You have to bring in all of these methodologies to try to pinpoint what we're looking for there.
Exactly, it's such a collaborative activity. Like, so many people are involved in this: pathologists, veterinary medics, zookeepers. And you're just trying to bring this data together. This is fantastic when it works, but it's a team effort for sure.
I'm going to bring in some more people from the audience to kind of mix in here. Rowan asks if you've looked at the mutation frequency among orthologs of known cancer genes.
That's a good question. So no, we kind of took just from like a genome-wide perspective. So we haven't looked in detail, because we don't get that many mutations within genic regions. Most of the genome is non-coding. So most of the mutations we're seeing are not in coding regions. So we haven't looked at that yet, but as we generate more data and we have kind of more saturation of mutations across the genome, we can start to look at, are there certain regions that are getting more mutations than others?
Great. And we got another question here from Carlo Maley. He's asking, if you drop humans as an outlier, is there still a very strong association of mutation rate with lifespan? That's
a good question. There is. We definitely checked that, and we did permutations of kind of removing pairs of species randomly and rerunning it. I can't remember those results. They might not be in the supplementary materials, but yes, we checked that because we were also worried, is it just explained by mouse or by human? And we still see this strong association.
So it's very robust to which species you include in the analysis.
It seems to be, because we were very, we didn't want to publish something that was wrong, and we were like, wow, this is quite a strong result. Is this real? So we tried to do as much as possible, permuting things around and seeing if it stayed there, and it seems to.
Awesome.
I think Carlo asked less questions at my PhD defense than he has today. But so pivoting from Carlo's questions. So now that we have so much of the comparative oncology work done, I mean, so many groups around the world, including ours, has just added to this portrait that we have of understanding cancer risk and the possible defenses across the tree of life. Where do you see this as we take a step back and we look at all these mechanisms, constraining somatic mutation, enhanced P53 copy number in elephants and other species? Looking at this, are these just mechanisms that are broadly important for somatic maintenance for these species that are going to live long or get large? Or do you think that cancer mortality in a population directly shapes these mechanisms to be specifically anti-cancer? Or is it a mixture of the both?
Yeah, it's a really good question. I don't know what the truth is either way or the other. I don't have a way to answer that at the moment. But I think, as James was mentioning earlier, we're increasingly seeing aging and cancer as potentially, through somatic evolution, is two sides of the same coin. So I guess it will come down to, in natural environments, what are those causes of mortality? How many animals are dying of cancer? And I guess, there's groups you're involved in who are generating that data from wild animals to try and learn what is the role of cancer in natural populations. And I think that is the data. So it's not always just me. I'm a geneticist, but it's not always genetic data that we need. So I think just having that data from wild populations about what is the role of cancer mortality in natural populations that's what's crucial for helping to answer that question.
Yeah, absolutely.
Great. Well, I think we should bring James back in, because I know James also has a lot of questions for you, Alex, and we want to dig deeper into some of these questions about aging. Welcome back, James.
Thanks. I've been in the backstage just itching to get back in because conversation. I really, in some of the issues that you raised, Alex, I think are so critical. And one of them being that it didn't scale to body size, which I find just as fascinating as the fact that it did scale to lifespan. Exactly. Because, as you mentioned, there's a huge variance in body size. And I think the difference between a blue whale and a mouse is close to 1 million-fold. So you presume they have, in other words, if on a per cell basis, they're accumulating the same number of mutations, that means that blue whale, its intestine has 1 million times as many mutations. So it's essentially had 1 million times, more chance of something going wrong. So just the per cell equalization of mutations during a lifespan doesn't really resolve that aspects of Peto's paradox. And just love to get your input on that.
No, exactly. Well, yeah, I mean, I think it feeds into our work was really on mutation rate, but the other work that like Josh Schiffman and other groups have been doing on these other mechanisms of specifically not necessarily controlling the mutation rate, but cancer resistance, where we see in elephants, 20, 40 copies of TP53. I guess, just from trying to make sense of the data that we have, that for large-bodied species, you have these, maybe evolution finds a better mechanism than just really reducing the somatic mutation rate, is being more sensitive to DNA damage. So just if you have a lot of cells you can afford to lose more cells, maybe if you have quadrillions as a whale, and you can just be more sensitive as a kind of like a gardener, just pruning anything that looks damaged potentially. But I mean, you do then run into these issues of, well, how do they not get stem cell exhaustion? How, is there a trade-off there? And again, I think it's just such an exciting time because I think we're starting to be able to get that information, being able to finally have the right tools to study these species that will help to generate the data to understand what are those mechanisms of cancer suppression. And does every large-bodied species, has it evolved a different solution? Or how many solutions to cancer suppression are there out there in the natural world? I think it's a fascinating question.
Yeah, I think the simple answer is lots. I mean, I think, clearly evolution has been, has taken many, ingenious routes to help us avoid both cancer and many other diseases. We often think about just cancer in old age, but pretty much our risk of everything bad goes up in old age. Heart disease, neurodegenerative diseases, so forth. And so that's just a reflection of natural selection. It's that waning of the pressure to keep us around, not so much a program to get rid of us. But
I want to jump in with a question from the audience, and then James, I promise we'll dig deeper here. But Michael Metzger he's curious about using the crypts and the fact that there was not an association with body size, and he points out further. He says if a bigger animal has more crypts that are of the same size, instead of having bigger crypts, would that potentially lead to a lack of association? And have you thought about potentially using other tissues to look at this? So is there anything kind of unique about using crypts that that could affect the outcomes?
Yeah, well, so, I mean, crypts are certainly a pretty unique, or that kind of crypt-like structure is not the most common structure. I mean, different tissues have very different structures that could seriously have implications for the way that mutations then spread with cancer. Crypts tend to be these kind of independent structures, whereas you have epithelial surfaces in the esophagus or the bladder, where a mutation can kind of spread much more easily if it has a proliferative advantage. And so, yes, there are potential ways that the mechanics could work differently in different tissues. And it's a great question because then I can talk about what we're currently working on, which is we were limited in the first study to using crypts because we had to use these kind of units where all the cells shared the same mutation because our sequencing wasn't accurate enough. But now we have these ultra accurate error-corrected duplex sequencing methods. We're now in the process of looking at way more tissue types across different species and beyond just mammals, where we can look at, crypts are very highly mitotically active cells. They divide a lot. So we also often think, well, could this just be explained by cell division rates? But we're now able to look at post-mitotic cells like neurons and muscle tissue. And so we're going to be looking across species. And that'll be really exciting to see, do we see the same patterns or does it vary? And then if we do see differences, maybe, yes, can we explain those by the unique kind of tissue architectures?
Yeah. Wow, so much exciting work coming up.
Yeah, it's I love it. I mean, it feels like a kid in a candy shop, right? There's just so many species, so many tissues, and we now have the tools to use them. And it's just kind of picking where do we start?
Yeah. Speaking of a kid in the candy shop, James, I'm going to let you get back to asking Alex questions.
No, I've got lots of them. So speaking of the different tissues, in some ways there already is indications from data from your lab and other labs that the number of mutations that accumulate in both a mouse and a human for the different tissues is relatively constant across different tissues. So I know that work from Inigo's group a couple years ago showed that the amount of mutations that you accumulate in a lifespan in a neuron is not that different than in a hematopoietic stem cell, for example. And so it does sort of seem like there's some sort of ceiling set by natural selection across species, but even within an organism across tissues, which again is amazing because you think about the different exposures that different tissues get, the different number of cell divisions, and every other variable you can imagine. It's amazing that there's been that sort of ceiling.
Yeah, no, I think a lot of the results are kind of shocking in a way, actually, and just like once you have the data, it's like, okay, well, this is the way it is, but actually, we're still trying to wrap our head around why do we see this limit. You know, it's not like an animal drops dead when it has 2,000 mutations. It's not that simple. So what are the ways, and why is there a similar number in a neuron as in a hematopoietic stem cell? What do those numbers really mean? It's kind of, evolution is telling us there must be probably a reason, but trying to understand all of the other layers of that. And I think it's going to be integrating a lot of different types of biology and information to really understand cell type by cell type what's going on.
Yeah, and you and I had an email discussion ages ago on this, but how much of this could be related to the sort of the control of germline mutation rates?
Yeah, so that's a great question. So whether it's just the selection for the germline and this is just a consequence of that, and it's certainly possible. I think we kind of lean towards that's probably not the explanation, because we do see different mutation rates between different somatic tissues. So it does seem like evolution can fine tune at that level, and you don't have to invest as much in somatic mutation. You know, I think evolution's able to decouple those. But we're in the process of now within the same animal measuring the germline and the somatic tissue mutation rates. So we'll get a kind of hopefully a clearer picture of how well do those two correlate.
Yeah, no, I agree. There's no way that's the only explanation, but I just wonder if it's a contributing factor.
It could be, sure. Yes.
So I guess another question would be is that you're seeing these effects on tissues, and it's mostly at a per cell level and over the lifespan, not as sort of a per organ cumulative mutation level. So could this relate as much or more to aging control as it is to, in other words, preventing aging during a lifespan? Aging in sort of the physiological decline, not just a chronological clock, as opposed to cancer. In other words, maybe this is more about aging than cancer.
Yeah, that's another great question. And I think that's probably the aspect of it that I'm most interested and excited about at the moment is what can this tell us about mechanisms of aging? And this is the kind of classical somatic mutation theory of aging, that all of our cells are just accumulating mutations that lead to decline in function over time. Every cell kind of breaks down. The number of mutations that we see, it makes us skeptical that alone is enough to cause aging, because most of these mutations aren't in genic regions. They're probably just kind of passive passengers. But we wonder, there's the kind of alternate theory that maybe you don't need it's not that all of these mutations are deleterious to the cell. Some of them actually increase the fitness of the cell and they trigger clonal expansions. Yeah. But they're beneficial to the cell, not necessarily the tissue or the organism. So could it be that you only need a small percentage of these cells to get these mutations that kind of will end up changing your tissue architecture in deleterious ways? And again, we now are starting to have technologies where we can start to explore that with targeted sequencing. So it's a whole new layer on top of mutations. We can now look at selection and that whole interplay of mutation selection, which is the classic kind of material of evolution, we can start to explore.
Absolutely, yeah.
I have to ask, what do you think of the idea, Alex, that some of these, expansions of cells that have mutations might actually be functional in that they sort of take up the ecological space, so that you don't get a more nasty mutation that might be more likely to sort of, disrupt like energy regulation of the organism, or cells that might be more likely to metastasize. They just, grow and take up space and maybe contribute to aging, but not as likely to contribute to cancer.
Yeah, like it's a better pathway for your cells to go down. Yeah, it's such a cool idea. And there is like some tantalizing evidence from the esophagus that we see more notch mutations that we see in tumors, but we actually see more of them in healthy tissue. So it kind of suggests that maybe there is this kind of pathway. And, then that leads to ideas for preventative medicine. Could you find ways to push our cells towards pathways that don't lead to cancer? That kind of, there's another dead end trajectory where they never transform.
Right. Or even good somatic evolution. I mean, exactly. Phil Jones's work, those notch mutations, if anything, seem to prevent more malignant clones from growing out.
Exactly.
So they actually might have a positive. But you could also imagine somatic evolution in our bodies that might be sort of tempering the aging associated decline. So I think we have to get away from the idea that all somatic evolution in our bodies is necessarily a bad thing. Of course, setting aside that, of course, somatic evolution in the immune system is a good thing.
Yeah.
But that even within our tissues, there's different flavors of somatic evolution, and some of it's malignant and some of it's not.
Exactly.
Yeah, that's a great point. And James, maybe kind of just expanding a little bit, like one of the sort of implications of this broad work that you're doing, Alex, is to sort of invite the community to think about somatic mutation in a deeper way. Think about, well, when is it functional? When might it decrease cancer risk? When does it increase cancer risk? How is it contributing to these other processes that have to do with sort of being multicellular organisms, right? Where like the cooperation of the cells is essential for it to function, but then mutations can sometimes lead to things that could be really detrimental, or sometimes the mutations might lead to things that stop worse things from happening. And sometimes the mutations lead to good things. Like James, you brought up somatic mutation in the immune system, right? Like the reason that we have an adaptive immune system is because our bodies and the bodies of lots of, animals leverage this capacity to somatically mutate to deal with the very, very rapid changes that can happen from agents of infection. So I think all of this does invite us to take that like step back and think of the broader perspective.
Absolutely.
Yeah. Alex, did you want to jump in with any other responses before we bring in Dawn?
Yeah, I guess just a quick thought. I guess I completely agree with what you're saying. And I guess part of that is the history of the field, that it was easier to sequence cancers before we could sequence normal tissue. So we start to think if there are mutations, they're bad. But it's just because we weren't able to look at the normal tissue before that. So we kind of primed ourselves to think somatic mutation, disease, but it's not always the case.
Yeah, they were sequencing normal tissue as the control, but literally, of course, the normal tissue is not clonal. Yeah. So you would miss out on the mutations. They'd see all this gamish, like in the little schematic you're showing. And so now with this either, small dissections like you guys did, or methods like, that you're referring to, the error correcting methods, you can actually look at a tissue and say, what is the somatic landscape of mutations? And that's just the power that even just a few years ago, we just did not have.
Yeah. No, that's a great point. And it's also just like a great point about sort of science and how to do science, right? You can't just assume that whatever you're going to choose as your control condition is like unitary in some way, right? You have to actually, have methods that allow you to look at what the variation is in your control condition as well as whatever your experimental condition is, because otherwise you don't really know what you're looking at, what you're comparing.
Absolutely.
Great. Well, I'm so excited to bring on our final discussant, Dawn Lehman. Welcome. Tell us a little bit about what brought you to evolution in cancer as a clinician. We don't have your audio yet, so gotta turn something on your end. Looks like right now you're muted, Dawn.
It was working before.
Oh, yeah. John, do you have headphones you can plug in real quick? I'm gonna remove some of us from the stream here to see if we can get. I think we're hearing like a little echo from you. Oh. Hearing something. Don, do you want to put any comments in the chat while we try to sort out the issue with the sound? Because yeah, it was working earlier when we did our test.
It's always the way it goes.
Yeah. You just sound very, very far away, and we can't even really understand your words. So if you post some things in the comments, I'll pull them in, or in the private chat. Alex, while we're waiting on Don's questions, is there anything about the sort of clinical side that you were interested in as you were sort of kind of trying to process, like, what do these results really mean?
Yeah, I mean, I think it's a bit of a challenge because these are like, the different species to start with, directly knowing what the immediate impacts are for the clinic. I think, overall, my main interest is ultimately you just need this better understanding of somatic mutation in normal tissues to be able to then make clinical advances, to really understand the full trajectory of cancer. And I think, from all my colleagues who are kind of applying these methods to humans, there's an enormous opportunity for things like earlier detection of cancer. So you can use non-invasive biopsies from urine or buccal swabs. And can those identify people where there's maybe a higher burden of somatic mutations that are then people who maybe we should monitor more closely because they're maybe closer to that initiating point of cancer, and understanding things like environmental risk factors or genetic predispositions through that lens of somatic mutational landscapes.
Yeah. Don, you want to try? Maybe we can get your question in. Oh.
Well, James says he has a medical question.
Yeah, let's bring James in too. Don, if you want to post, you can post in the private chat. That's part of the StreamYard interface here, or you can post in the comments on YouTube, and I'll pull it in.
Yeah, so I'll go and ask one. So sorry, Don, about the technical problems, but so one of the things that I think it would be fascinating, and love to get your comment on this, and whether you guys have any plans for this, would be to, ask whether, not just sort of understanding, what the landscape is, but particularly once we start getting a better understanding of when a mutational landscape might be a dangerous mutational landscape. And I'm using the word dangerous because I'm not necessarily referring to cancer. Maybe it's something that contributes to tissue decline or whatever. But then you could ask, okay, do people that exercise have a more benign looking landscape and so forth? And so maybe by looking at behaviors and lifestyles that we know are good for lifespan and not getting cancer, and comparing their mutational landscape to those of smokers who never get off the couch, might be able to get an idea of what a good landscape and a bad landscape is.
Yeah, no, absolutely. We don't. I mean, so something Sanger is very good at is large scale, enormous sequencing projects. And I think to answer those kind of questions, that's the ideal scenario, situation you want. Is we're hoping to do these large scale studies where we can look at people with different risk factors, different lifestyles, and then we have this idea of somatic mutations in normal tissues, but what is normal? And as you mentioned, there's this whole variation of different lifestyles that people have and different exposures. So building up a much more nuanced, detailed picture of how landscapes relate to those different factors. And I think you're right. Then hopefully that could be a link between some of those things like obesity or exercise or stress, chronic stress. How do those influence landscapes? Do they do it in different ways, similar ways? And yeah, I think that has enormous potential for what it can tell us about monitoring people better, and also understanding how does obesity increase cancer risk or exercise. And I think clonal evolution is probably a likely mechanism that we'll see that will explain some of those, how those lifestyle factors influence risk.
Exactly. Then we can start modeling those mutational changes, whether it's in cell culture models or animal models, and asking, do the ones that are associated with exercise, maybe they don't disrupt function or lead to malignancies, but the ones that are associated with the couch potato smoker do. Yeah.
No, absolutely. And again, we now have the methods that we can start to go about addressing these questions. So I think the next few years are going to be extremely exciting.
Yeah. Now, you could even look at if there's certain kinds of mutations that are more likely with certain risk factors, right? Like that could be really fascinating as well.
Absolutely. Yeah, I think like Carlo's question was mentioning earlier, like things like oxidative damage, are those kind of mutational patterns more likely with certain habits that then lead to certain cancers?
Absolutely.
Seems like a good use for machine learning.
Yeah, no, absolutely. I mean, I think these mutational signature analyses, we kind of look at quite a simple level of trinucleotide spectra, but with the machine learning capabilities, you can start to see relationships potentially with like 3D structure of the DNA, these larger conformational changes that we just can't really pick up. But with machine learning, and enough data, you'll start to pick up these other associations potentially.
Yeah. I think enough data is the key term there.
Yeah, exactly. Yeah.
Great. Well, I think we should bring Zach back in because he's backstage, and I'm sure that he has some thoughts that he's been sitting on. So
Thanks, Peto.
Yeah.
Alex, I saw on Twitter, and no one's congratulated you yet, but starting your research program now at the University of Cambridge. Congratulations on that. What from what you've learned from like this monumental paper that you've put out and research interests of your own, which of these themes you're going to carry over and define your research program going forward?
Oh, thanks for a very nice question. So yeah, I started on Monday. And so yeah, my group will be focused on leveraging this huge diversity of lifespans and cancer susceptibilities across the tree of life and applying these kind of new tools that we have. So as I kind of mentioned, we'll be looking for the species with the longest lifespans or the lowest cancer resistances, looking at both the mutation rates, which we've kind of started, and then on top of that, looking at the selective landscape. So we know that somatic mutation rates in mammals correlate with lifespan, but do the rates of clonal expansion, do the clonal landscapes also correlate with lifespan? And then the other thing I'm very kind of excited to try and look into a proof of principle is, can we leverage this information from somatic mutations in non-human species to apply to environmental monitoring? So we know that not all carcinogens are mutagenic. They can also increase cancer risk in other ways. And so I'm very interested in kind of taking a polluted ecosystem, looking at different species within that ecosystem with somatic mutational techniques to see, do we see mutational signatures from those pollutants? And do we see changes in the clonal landscape that could explain the kind of negative effects of different pollutants? And so Charlie Swanton has some really exciting recent work showing that kind of urban pollution from cars doesn't necessarily increase mutation rate, but you get this change in the local tissue architecture in the lungs. It promotes expansion of pre-existing mutations. So hoping that we can then apply that to different species.
That's fascinating.
Awesome.
So we have a few questions from the audience that I'd love to bring in here. So Docs Talk Shop asks, when you do get your first whale, what do you think you're going to learn from that about possibly avoiding cancer?
Well, that's a great question.
And what are you going to name it?
So first of all, I think we'd just like to confirm in some of these cetaceans like bowhead whale, do they have lower somatic mutation rates, which would kind of confirm this idea that there is this relationship that extends beyond human lifespan, that humans don't have the most efficient somatic mutation that we could have, that mammals are able to lower the somatic mutation rate. So there may be mechanisms by which we could do that. And kind of as well as that, we can look at the clonal landscapes again, so seeing how those vary with lifespan. And I think, there's already some exciting data from Vera Gorbanova's group where they've taken bowhead whale cells and shown that they have more effective double-strand break repair. So I think, a key question, we've looked at somatic mutation rate, but in our studies, we've just looked at these kind of small single nucleotide variation. And we'd really like with long read sequencing to start to look at other scales of somatic mutation. So there's structural variants, which one could hypothesize have a larger effect on the cell because you're rearranging much larger amounts of DNA every time you have a large structural variant. So I think with these kind of large cetaceans, it'll be fascinating to see, have they really focused just on structural variant repair, or do they also have less kind of smaller somatic mutations? Because that might give us a clue about what's really critical for fixing to reduce cancer risk. Do you have to reduce all somatic mutations, or should we really be focusing on those larger kind of chromosome level variants?
We've got Animal Hour going on here. Carlo's asking, Greenland sharks?
We don't have any data yet. One washed up on the UK shoreline last year, very kind of it. So we have one Greenland shark, and through collaborators, we're going to be getting 7 or 8 more. So that's top of the list. So hopefully in 6 months' time, we will have Greenland shark data.
Well, Alex, as you touched on this, as you're trying to go through the sample acquisition, especially on these super rare species, the Greenland shark washing up on shores was a stroke of brilliant luck. But what's more important to you as you set out on this, making sure you have ways of accurately identifying their age, or just getting them and doing what you can with them?
It's a bit of both. At the moment, because we know so little, we can kind of be selective. And I'm trying to look at species where we can get the best possible set of individuals of multiple age, and we know the age.
Right.
But I am exploring, there are ways with epigenetic clocks or other mechanisms you can try and get that age data. But I think at the moment we can try and be kind of fussy and be like, we just want to make sure we have the cleanest data possible. So trying to really go down. But having said that, Greenland shark is such a fascinating species. But that's why we make an exception, and we'll just look at it because we can still get a rough age estimate based on size. And given if we think it's 300 years old, just knowing what the mutation rate, give or take a few decades, is still going to be informative.
Yeah, absolutely.
Okay, We've got a few more questions from the audience. We have Michael Metzger asking if there are any long, short-lived, closely related animal pairs that you really are dying to look at.
Lots, but right now with Dario Valezzano's group in Germany, we're going to be looking at somatic mutation rates in these killifish. So they're one of the shortest lived vertebrate species, and within killifish you have kind of orders of magnitude difference in lifespan between closely related species. So some of them only live for a couple of months, and then some can live, I think, for a couple of years. And they're in very close proximity geographically, kind of just depending on the ecology of the water systems they live in, how quickly those pools dry out kind of shapes their lifespan. And so I think that's a really nice model system where they can keep them in the lab, and we can really see, are those mutation rates scaling with lifespan? But yeah, I'm always welcome people to reach out to me. If you think anyone on this call, this is a great species pair that would be ideal, let me know, because there's just such incredible natural diversity out there, and there's so many great systems for different kind of questions we want to look at. One other great one we want to look at is ants, so eusocial insects, where you have a queen and a worker, and the queen, they share the same genome, but the queen in some species can live 20 to 30 years, and the workers are living less than a year. And so trying to understand what are the maybe epigenetic mechanisms, is there enhanced DNA repair in the queen, and kind of looking at these species comparisons, as the questioner mentioned, can be really informative when they're closely related, to try and understand, to try and identify those mechanisms of somatic mutation repair.
All right, we have another animal question. Barrel sponges that can live 11,000 years. Have you looked at those or thought about looking at them?
Yeah. The sponge is definitely on the list. If as many grants as I can get, the more species I will. But again, I think another thing is, like, now these methods are published, so I really hope that lots of other groups will get involved, because the whole tree of life is out there practically unexplored, and these methods aren't that hard to apply, really. Now that the pipelines are being released and are out there, any lab with a sequencer essentially can do a duplex sequencing protocol and start to look at somatic mutation rates in these species. And the more data we can get from the more species will make it just so much more easy, using the power of comparative biology, to learn about what are those life history traits and what are those mechanisms that are recurrent in evolution. I'll just pin this down.
We do have a methodological question here from Jamie Modiano asking whether there's sort of differences here in the clone seen in young individuals, and how the chip overlay on the rate of somatic mutation may be affecting those results. I don't know if that's something that you could respond to real quick.
Yeah. So we have very limited data on clonal evolution at the moment in these species. It's really just looking at the moment in somatic mutation rate, but it's ongoing work. So we're getting the bait sets in now to start to be able to look at clonal evolution across different species. But that is a kind of a key question to us: is how does the kind of just raw mutation data relate to the clonal processes of, like, clonal hematopoiesis in different species? Do they scale with each other, or are they completely different relationships? And which of those are important for actual mechanisms of aging? I mean, it's pretty clear that maintaining genomic integrity matters for aging, but is it because of mutations that are deleterious to the cell, or is it because they trigger these clonal landscapes, and that's what damages the cell? And I think that's a really fascinating debate. Don't know if it's going to be probably a synergy of the two, but trying to actually quantify that is kind of I think a key question.
Great. Well, I'm going to invite James on to give any final thoughts, and then Zach, and then we'll be finishing out our hour. James, any last thoughts you want to share with Alex and the rest of us?
First, it's just been super fun. I mean, and I'm like dying to see the killifish result. To me, that's one of the most fascinating comparisons, because the amount of evolutionary time between the short and long-lived ones is not that long, and yet they vary, like you said, by 10-fold in terms of their lifespans. So I think that's going to be a really fascinating comparison. I hadn't even thought about the ants. That's also fascinating. But one thing you may want to think about with the killifish, and I don't know if you're planning this, is that there are different pairs, right, that are on different, in other words, the shorter lifespan has evolved multiple times.
Yeah.
And so you could actually do the experiment on one branch and do the same experiment on another branch.
That's the plan. Yeah, exactly.
Oh, that is. Oh,
yeah. But yeah, no, I can't wait for the results. As soon as we get the permits, we'll get it.
You get to go fishing.
Yeah. Well, James, thanks so much for sharing all of your thoughts and expertise on this live stream. It's been awesome having you guide us through everything.
Oh, no, it's been fantastic.
Yeah,
Thanks for serving as MC.
My pleasure. Zach, how about you? Any final thoughts?
Yeah, I mean, I'll echo James. I mean, even on the comparative oncology side, we're always fascinated in these natural experiments, trying to find these phylogenetic pairs that have vastly different cancer rates. I think broadly, I mean, I was so excited with your paper and bringing in both the people interested in comparative cancer analyses and the comparative aging as we kind of merge fields and approaches and methods. I think it's going to be really exciting. I'm really excited to see what you're going to do now that you're on to the next step of your career, Alex.
No, thanks very much. Yeah, I would just want to thank everyone here. I mean, I wouldn't be in this field if it wasn't for this community. So I was working on population genetics. I got invited to illustrate one of these iSeq conferences, which are great conferences everyone should go to. And I was like, wow, there's this whole world of somatic evolution that we can now start to study. And I was like, I need to study this. And I feel like this field is really just at the tip of the iceberg stage. And it's going to be so fascinating over the next years to see what we discover from studying cancer rates, somatic evolution, clonal landscapes across different species.
Absolutely. Zach, thanks so much for being on with us. Great to have you. And
thanks, Athena.
Yeah. Now, Alex, I'll just say, I mean, to me, your work is so exciting for a lot of reasons, but especially because, you know, I think about multicellular bodies as these like amazing examples of cellular cooperation, right? It's like 30 billion cells that somehow manage to cooperate and coordinate their behavior for, you know, decades often, at just a massive scale. Like, you know, we humans could never cooperate on that level with each other, right? And, you know, and then thinking about this like mutation process, not just as something that can lead to cancer, which is a very sort of specific, clear, and often fatal breakdown of cooperation, but how it can lead to these, you know, other processes that might just kind of, you know, slow down the ability of the organism to function optimally, that might contribute to poorer health. And then, you know, it kind of opens up this whole world of thinking about health from the perspective of somatic mutation, right? And somatic mutation and sort of cellular cheating, maybe as this broader mechanism that might be at the root of many different challenges that we have in health, whether it has to do with metabolic regulation, dedifferentiation, any of those things. So to me, it really opens up a whole new lens for looking at those issues.
Yeah, no, absolutely. And I think we're very fortunate that now we've mentioned a few times, we have amazing tools now that we can actually quantify those things so precisely that that's really essential to then learning about all these different processes that you mentioned.
Yeah. Alex, any final thoughts you want to leave us with, or teasers for what we can look forward to coming out of your work?
I mean, I think I've mentioned all the things I'm enthusiastic about, but yeah, I think so many species that we're in the process of looking at. We're starting to get preliminary data from neuronal cells in lions and killifish, zebrafish, and I can't wait to share the results hopefully in the next year or so to kind of help just like, I feel like there's just so much of this picture that hasn't been painted yet, and just kind of generating that data, it's just going to give us such a richer understanding. And I can't wait to see results from other groups looking at similar processes and get our heads together. It just takes a whole community of ecologists, evolutionary biologists, geneticists to understand this topic. And there's so many different ways that people can contribute. So I can't wait to see the field to keep growing, really.
Absolutely. Oh, and let me just say, everyone, if you are not already following Alex on Twitter, Alex, what's your handle there?
The handle is @ATJ_Kagan.
Okay, because Alex has the most awesome illustrations that he puts on Twitter about his work, and I just found that, you know, getting to see things in such a visual way has been awesome for me, for understanding your work and other work that's happening in the field. So thanks for adding that dimension to the field.
Pleasure.
Thanks for being with us, Alex. I hope that we'll get the chance to talk to you more about your work and see you hopefully at iSeq in May.
Yeah, look forward to it. I love that meeting. I'll be there.
Yeah. So shortly, we're going to be sending out some more information about that. It's going to be May 8th through 10th at the Wellcome Trust Genome Center in Cambridge in the UK. So I hope we'll see you there, and I hope we'll see everyone else there who's been joining us for this live stream. So Alex, thank you. And thank you to everyone who's joined us today.
Thank you.